Structural organization of proteins. There are 4 different levels of organization of proteins: primary, secondary, tertiary, and coming to primary structure of proteins. This represents linear sequence of amino acids joined by a peptide bond. Now the importance of primary structure is that this structure determines a higher level of protein structure and also determines the biological activity of the protein. So the main force that stabilizes the primary structure is peptide bond. The characteristic feature of a peptide bond include it's a partial double bond. It is rigid and planar, no free rotation around the bond. It is uncharged and it resists denaturation. Coming to naming of the peptide bond, free amino group is always written on the left side and the carboxyl group to the right side. For example, let's assume this tripeptide which is made up of N-terminal valine, glycine and C-terminal leucine, and this is read as valyl glycyl leucine. Clinical significance of primary structure is that, for example, in sickle cell anemia, so there occurs mutation at beta 6 position of hemoglobin where glutamic acid is replaced by valine, and this decreases the oxygen carrying capacity of erythrocytes, and erythrocytes get converted from biconcave shape to sickle shape. Coming to secondary level of organization of proteins, this refers to relationship of amino acids that are 3 to 4 residues apart in the linear sequence. Types include alpha helix, represents the spiral structure, and each turn of the helix, alpha helix contains 3.6 amino acid residues, and the spacing between each amino acid is around 1.5 degree angstrom. The next type is beta pleated sheet, has got a sheet-like structure and the polypeptide chain is fully extended, and the distance between adjacent amino acid is around 3.5 degree angstrom. It can be either parallel, example flavodoxin, anti-parallel arrangement, silk fibron, and mixed, seen in carbonic anhydrase enzyme. The next type is the beta turns of the motif. These are super secondary structures, and they usually they provide linking between the alpha helix or the beta pleated sheet. Example, beta hairpin, where the two antiparallel beta strands are joined by tight turn of amino acids. Helix loop helix, two alpha helices are joined by loop of stretch of amino acids, and zinc finger motifs mainly seen in DNA, where two beta strands are linked alpha helix. Coming to tertiary level of organization of proteins, this represents the relationship of amino acids in the secondary and the primary structure that are placed far apart, and this also contributes to three-dimensional structure of the proteins, and thermodynamically this is the most stable structure of the protein. The stabilizing forces include the disulfide bonds, hydrophobic interactions, ionic interactions, hydrogen bonds, and van der Waals forces. Two types of tertiary structure are there. One is the fibrous proteins, which mainly serve the structural roles. They have simple repeating elements of secondary structure, example collagen. Next are the globular proteins. They have more complicated secondary structure, mainly containing several types of secondary structure in the same polypeptide chain, and the folding of this globular proteins provide structural diversity necessary for proteins to carry out wide variety of biological functions inside the human body. For examples for globular proteins include the enzyme transport proteins, motor proteins, regulatory proteins, and immunoglobulins. The clinical significance associated with tertiary structure of proteins is the prion disease, which occur because of abnormal protein folding. So the examples of prion disease includes the Creutzfeldt-Jakob disease in humans and mad cow disease in animals. The last one is the quaternary level of organization of the proteins, and this mainly represents aggregation of the polypeptide chains or the aggregation of the subunits to form one functional protein, and the protein loses its function when the subunits are dissociated. So this is a characteristic feature of quaternary structure of proteins. The forces stabilizing include the hydrophobic and the ionic interactions, hydrogen bonds, and the weak van der Waals forces. Coming to types of quaternary structure, they can be homodimer with two copies of the same polypeptides, or heterodimer, two different polypeptides. Examples include hemoglobin, made up of 4 polypeptide chains, 2 alpha and 2 beta. Immunoglobulin contain 2 light chain and 2